by MD and CEO of Arovella Therapeutics, Dr. Michael Baker. Today, Michael will give us a presentation and then we'll take questions from the floor. So feel free to pop in your questions whilst Michael is presenting if you'd like. Thank you so much, Michael, for joining us. I'll hand it off to you.
Wonderful. Thanks, Caitlin. Appreciate the time and you putting together this event. And of course, thank you very much to everybody else for taking the time to listen in. So my name is Michael Baker. I'm the CEO and Managing Director of Arovella. We are a biotechnology company listed on the ASX under the ticker ALA, and I'm looking forward to taking you through our story. So there's the disclaimer. And I wanted to start with a snapshot of some of the metrics of the company. So we're currently valued at about 17.5 million. End of March, we had $8.1 million cash in the bank. So leaves us with a pretty modest enterprise value, and I think that's largely off the back of a pretty ordinary run for biotech companies in general, but certainly the way we see it with the technologies that we've acquired from Imperial College London, as well as MD Anderson Cancer Centre and the team that we've built around those technologies, we do see that there's a fair bit of value creation for us on the horizon. And in terms of our shareholders, we're delighted we completed a placement earlier in the year. Very happy to welcome Merchant Group to the register. They've had a number of wins in the biotech sector and they continue to be in the process of raising a dedicated biotechnology fund. So good to have them who understand the sector quite well, and of course the investment horizon for these types of technologies. And we also have a number of high net worths and small institutional investors on the register, but of course we're always looking to welcome more investors to the register that understand the risks and the investment horizon for a biotechnology investment, but of course the returns that can be achieved in this sector. And so what it is exactly that we work on, in a word, it's cancer, and it's a huge medical health problem. And I think everybody certainly listening on the call, I'm sure has been impacted in some way or another by this devastating disease. And that's largely due to the prevalence. In 2020, we saw around 19.3 million new cancer cases, as well as in the same year, almost 10 million deaths. So it is an enormous health issue. We continue to make advances, but of course, there's a lot of room to do better when we see those staggering figures. And in terms of the commercial outlook, it is an enormous market, not surprisingly, and it's expected to reach somewhere between 300 and 400 billion by 2030. And of course, as I said, it just means that the problem from a healthcare perspective, there's a lot of opportunity to do great things, but of course, there's a lot of opportunity commercially as well. And so more specifically, the type of therapy that we actually work on is a type of immunotherapy called a cell therapy. And they essentially, we're using the word revolution here because they have changed the way we think about cancer treatment. And as of the middle of this year, there are actually 6 approved, what we more affectionately refer to as CAR T, with the T standing for the immune cell that's of use. It's a T cell immune cell. And they've been approved to treat a range of blood cancers. But as I said, it's the impressive cure rates that we've seen for these therapies that has really heralded in a new way of how we think about cancer treatment. And so in February this year, there was a series of articles published where some of the patients that had actually taken these therapies experimentally 10 years ago, they're actually still cancer-free. And so that's a nice position, as I said, where we're using the word cure and cancer in the same sentence. And more recently, a couple of months ago, there's also a series of articles following up on the first pediatric patient to receive these types of therapies, Emily Whitehead, and again, 10 years on, she's still cancer-free. So it is a very exciting time for these types of therapies, and certainly that's what gives us a lot of motivation moving forward. And so how are we different? As I mentioned before, these therapies initially were referred to as CAR T, and that's because of the immune cell that they actually rely on for the therapies to be produced, whereas we're working with a different, still naturally occurring immune cell. It's called the invariant natural killer T cell, or iNKT for short. And we think that that has a number of advantages over the conventional cell therapies. And I'll talk through those in a moment, but first I'd just like to talk about what the therapy looks like, so to give people an understanding of what we're dealing with, but again, why it's so exciting. And so what we actually do is we start from a healthy donor here. We collect the the immune cells, or sorry, the blood from that donor, and then we select out the immune cells of interest. As I mentioned, in our case, it's the iNKT cells. And then we genetically reprogram these cells to produce something that we call it's called a chimeric antigen receptor, but we call it CAR for short, and that's where we get the CAR iNKT. And the end of these little CARs, what they actually become is like a guided missile dragging the immune cells towards the cancer cell, and when they find them, they trigger their destruction. And so after the genetic engineering stage, we grow the cells up into very large numbers in the billions. They then get frozen down and stored and ready for use, so that when a cancer patient presents and is eligible to take the therapy, we're able to take them out of the freezer and administer them to that patient. And so one of the biggest advantages of our iNKT cell therapy is really around the manufacturing. So for the conventional T cell therapies, for those that have been approved, they can't start from a healthy donor. They must come from the patient's own blood. So they take the patient's blood, collect the immune cells, and go through this process. And what ends up happening is it becomes a very expensive, very logistically challenging process. And what ends up happening is that in some cases, some cancer patients, their disease is too aggressive. So they don't actually last the 4 to 6 weeks that it takes to manufacture the therapy. And as I said, of course, the resultant cost of a therapy like this, because it has to be made for every single patient, is quite high. So we're aiming to have a product where we don't need to do that. We start from the healthy donor, we freeze it down, and it's ready for use, which simplifies the supply chain. Of course, will bring down costs. Now, in addition to that, iNKT cells are one of the most potent immune cells, so they're very effective at killing cancer cells, which I'll show in a moment. They also activate other components of the immune system to join in the fight against cancer. And we've got data to show that for tumours that produce something that's called CD1D, they actually perform better than the conventional cell therapies. So in terms of our pipeline, we have 2 major products in the cell therapy space that we're looking at. The first is ELA-101. It's in the preclinical stages, and we're targeting a form of blood cancer called lymphoma that produces what we call CD19. So that's a technology that we licensed from Imperial College London. And the second is ELA-104, where we licensed that technology from MD Anderson Cancer Center, and we're using that to target another form of blood cancer, multiple myeloma. But based off the data, we could also be looking to tackle solid tumors as well. So for both of these therapies, they are in the preclinical stage, and the key next step for us is to take them into clinical trials in humans. Now, we also have an oral spray platform. I won't spend too much time on it today, but we've got a series of drugs under development where we take them from solid dose form and convert them into oral spray. I'll just mention our lead candidate for this platform. It's called Zolpimist for the treatment of short-term insomnia. We have partnered with Teva for that, who are the world's largest generics manufacturer, and Starter, who commercialized that in Australia last month. So the idea is we continue to work with Teva to get regulatory approvals and commercialize across South America. Now, I'd like to talk more about the iNKT cell therapy platform. I gave you an idea of the process, but I'd just like to talk a little bit through about how it works. And as I mentioned, that was licensed from Imperial College in the middle of last year, and that's the inventor and one of the senior postdoctoral researchers that runs the research in the laboratory. So I'd just like to talk about how it actually works. So just to make it crystal clear, so we have the iNKT cell. They already contain what we call an invariant T cell receptor, or ITCR for short, that can recognize and trigger the destruction of cancer cells. This is unlike T cells, the conventional therapies. As I said, we genetically engineer them to produce a CAR. Now, we can change the end of that CAR so that we can utilize that to target a range of different cancers. And then once we give them to the patient, what we have is the ITCR recognizes what's called CD1D on the cancer cell, and the chimeric antigen receptor recognizes what we tell it to on the cancer cell. And so once that happens, we get the destruction of the cancer cells, and other components of the immune system come in also to eradicate the cancer cells that are present. And so just to show you, again, these are the preclinical studies that were published in the prestigious journal Cancer Cell, just to show you what it looks like in an animal model and why we're so excited by it. This is a series of experiments, but what we know from them is that the iNKT cells have faster killing of cancer than the conventional cell therapies. And so what we're looking at here is the top two rows, just the underside and the backside of the mice. And this is after the cancer has been given to the animals. So anywhere you see the blue, the yellow, or red or green, that's actually the cancer cells in the animal. And then what we're looking at on the bottom two rows is the animals, the same animals, but now after 3 days of treatment. And so in the first three columns here, we see this is essentially no treatment. And we see that the animals have all continued to get worse in terms of the cancer. This is just the immune cells without having the chimeric antigen receptors or CARs, and we still see that they don't perform that well. And then when we look at the T cell with the CAR, we still see after 3 days that there's quite a bit of cancer cell remaining in the animals. But when we look at our therapy, it's very, very clear that after 3 days, the tumor cells have been eradicated. And so what's really important is, great, we see very fast clearance, but what does that mean in terms of overall survival for the animals? And so that's what we do here. We look and see how long the animals are alive for. And if we go out to 90 days, you can see that, and sorry, on the horizontal axis, we're looking at time, and on the vertical axis, how many animals are still alive, with 100% being at the top. And so for the T cells, we see that there's about 60 to 65% of the animals are still alive, versus the iNKT cells, it's in the order of 90 to 95, which demonstrates better survival using our therapy as well. And so for as I mentioned, the key next steps for this are to manufacture the drug at a quality level suitable for clinical trials. And we've already selected the key component manufacturers for this product, and now we're going through the process of setting up the manufacturing at our cell therapy manufacturer, Cugen. And so I just wanted to talk more about the second target that we've got. It's called ALA-104. So this is a technology that we're licensed in from MD Anderson Cancer Center, which is regarded as one of, if not the best cancer center worldwide. And as I said, it was developed there as what we call a monoclonal antibody. And we can use this technology and incorporate it as a chimeric antigen receptor, or CAR. And so that's the work that's been done to date from the inventor. He's actually used it as a CAR, and he's done that work in T cells. So that's the conventional type of cell therapy that I mentioned a moment ago. And what he's done is shown how this performs in multiple myeloma, a form of blood cancer, as well as 3 solid tumor cancers, and all using animal models, and demonstrated excellent activity for this particular therapy. So what we're going to be doing is confirming some of the earlier work that he's done around target specificity and selectivity, and basically take that CAR and put it into our iNKT cell platform. And initially we'll be targeting multiple myeloma, but we do think that there is scope to target some of those solid tumors as we move forward. And again, just to give you a snapshot of some of the data that's been generated to date for this particular product, we're looking at 3 different forms of the blood cancer called multiple myeloma. And what we're looking at here is, again, these survival curves, and on the horizontal axis, it's how long the animals are alive and how many animals are alive at that particular time. And in the black line, what we see, these are all the control animals where they actually haven't been given anything that's going to prolong their survival, and they die between 30 to 40 days in these models. And what we're looking at in the red line is the technology from the inventor, again in T cells, but what we see is out to 50 to 60 days, these animals are still alive, and again, demonstrating that this is having a very significant and great response against these particular forms of multiple myeloma. Now, what we know about multiple myeloma is it also is a cancer that produces CD1D. So we think that by putting this technology into our iNKT cell platform, it will make our cells dual targeting, and so we'll have a better performing product. And as I mentioned, there's great animal data already to demonstrated in cancer models depicting lung, pancreatic, and triple negative breast cancer. So the key next steps for these therapies are, firstly, for ALA-101, as I mentioned, to coordinate the manufacturing of the product, which is transferring the technology and the manufacturing process to our cell therapy manufacturer and completing what we call viral vector. That's how we get the CAR into the cells, the iNKT cells, and then completing the manufacturing of the therapy. And as we move, we'll complete any necessary investigational new drug or IND enabling studies that are requested before ultimately getting regulatory clearance so that we're able to commence a phase 1 clinical trial in humans and then to dose the first patient in a phase 1 clinical trial. For ALA-104, as I mentioned, a lot of work has been done looking at the safety and specificity of the technology already, but we would like to confirm that on our end. And while we're doing that, we'll combine the CAR technology with our cell therapy platform so we can test the activity in the cancers that I mentioned, multiple myeloma, and then expand into breast cancer, lung cancer, and pancreatic cancer. And once we're comfortable with that data, we'll take the process that we've learned from this therapy to define the manufacturing strategy and then produce that therapy for clinical trials. So I think it's worth touching on the cell therapy commercial activity landscape because it has been quite a busy area and it continues to attract a lot of investment, both from a transaction point of view and capital raising point of view. And since 2017, when the first 2 therapies were approved, we've seen a number of transactions in the billions, but now we continue to see a lot of strategic collaborations and partnerships between large pharma and smaller biotechnology companies. And this is even for companies that are in the preclinical stages, so before they put their therapies into humans. And on the capital raising side, we've also seen some very large, several $100 million capital raises, and we continue to see some large raises even over 2022. Again, just reiterating, this is still an area where people are quite excited by the opportunity. Again, going back to what I showed earlier, because we've seen such robust data and we're starting to use the word cure for these types of therapies. And so in terms of the global cell and gene therapy market, it is expected to reach around 13 billion by 2026. But no doubt it will be a very big market moving forward and a key part of the cancer treatment regimen moving forward. Now, more specifically, I wanted to talk about the iNKT cell therapy space. So if we look at T cells or the conventional types of cell therapies, there are hundreds of companies and hundreds of clinical trials ongoing. So it is quite a busy area and a busy field. Whereas when we look specifically at the iNKT cell therapies, there are only a handful of companies working on this platform and only a handful of clinical trials. So we do find ourselves in a far less crowded space, which is an advantage for Arovella. But nevertheless, over the last, say, 12 to 14 months, we've seen quite a bit of commercial activity for all 4 of the players that I've listed here. PO Bio raised 52 million in May last year, and as I mentioned earlier, they entered into a strategic partnership at the preclinical stages, so they're not expected to reach clinical trials in humans until the end of calendar year 2023, but they already have a strategic partnership worth up to $875 million with Kite Pharma. And Cure, they're a little bit later stage. They have a phase 1 or 2 phase 1 programs already. They were acquired for 185 million in May of 2021, and that's the total deal value. Mint Therapeutics raised 40 million in October last year. They've been, like most biotechs, they've suffered through the recent downturn and currently valued at 43 million US, but still stands quite a bit above Arovella, which is valued at, as I said, just about 17.5 million. And for Arovella, of course, what we've seen over the course of the last, say, 12 months or so is that we've licensed in a new cell therapy platform from Imperial College London, a top 10 university worldwide, and also the DKK1 technology from the MD Anderson Cancer Center, which is one of the world's best cancer centers globally. And so I think really behind the technologies that we're working on, equally important is to have an outstanding board and management group, and that's what we've assembled over the last 12 months following acquisition of the technologies. So we're delighted to have Dr. Elizabeth Stoner as our interim chairperson. She was formerly an assistant professor at Cornell University in the US, followed by senior clinical development roles at large pharma like Merck, and now she's the executive partner at MPM Capital, which is a well-known life sciences venture capital fund based in the US. So myself, I'm a PhD biochemist by background. before moving into drug development and venture investing before joining Arovella as the CEO in 2020. Dr. Deborah Barton also has a wealth of experience at a number of cell therapy companies, and her last two have been in the C-suite. So we're delighted to have somebody with the clinical development experience like Deborah for cell therapy specifically. Gary Phillips, we appointed to the board just this month. Delighted to have him join. He's very well known for his role at Pharmaxis as the CEO for the last 10 years. So again, a wealth of information across the life sciences and drug development sector. And these two individuals, Dr. Sandy Buchanan and Dr. Minnie Barathan, were recruited really to drive the development of our therapies. Both are based in North America, in the US, I should say. And Sandy is our VP of Manufacturing and Quality, which is really important because it is a key part of getting these therapies into clinic. And Dr. Minnie Barathan is our VP of Development and Translational Medicine, again, with more than 15 years of experience in developing these types of products, and will play a key role in taking them from the preclinical stages and into and through clinical trials. And so just to summarize the company, I think it's clear that we do have world-leading partners in Imperial College London. As I mentioned, it's a top 10 university worldwide. That's where our cell therapy platform was originally developed, and we now have our new DKK1 targeting technology from MD Anderson. And so those two combined give us what we call an allogeneic platform. So that's that concept of off the shelf, or being able to give the therapy from a healthy donor to a patient with two exciting targets. And DKK1 will be the only company in the world that's working on this particular target. And as I just highlighted, we have a world-class team with a lot of experience coming out of key markets where these therapies have been developed, and a lot of experience, particularly with cell therapies, which gives us a big advantage. One thing I didn't talk through today, but what we continue to do is look to acquire new technologies that we think can either synergize with our current platform or even give us new areas to focus on. And again, we've got a lot of experience and VC experience in our team, which helps us to be able to do that. In terms of our decision making, it's very data driven, and that's what we look for. We believe that when you've got world-class data, that underpins great data, and that underpins great therapeutics. So we're very much a data-driven organization. And then for all those reasons, as I said, we're the only ASX listed company that's working on an iNKT cell therapy platform, and we are the only company, as far as we know, that's working on a technology like the one we've got that's targeting DKK1 producing cancers. We do see that there's a lot of upside for the company, and certainly looking forward to welcoming new investors to the register. And with that, I'd just like to say again, thanks very much for the opportunity to present, and I'd be delighted to take any questions.
Thank you so much, Michael. What you guys are working on is so revolutionary, and I think the case studies that you were talking about in the beginning really reflect that. So it's amazing. We've got a few questions already, but to those that came in late, feel free to pop in your questions and we'll go through them. So firstly, how long does the CAR-iNKT cell therapy process actually go for? How long does it take for the manufacturing and the treatment, I guess?
Yeah, and that's a really great question. So if you're doing it with the conventional type of therapy, it can be in the order of 4 to 6 weeks. And the time that they refer to for those is vein to vein. So you're talking about the time it takes to take a patient's blood and how long it takes to genetically modify, grow up the cells, have them shipped all over the world, and then back to the patient where they're injected again. And as I said, that's about 4 to 6 weeks. So it takes too long in some cases, and patients actually with aggressive disease won't last that journey, unfortunately. And so what we have for our therapy is the ability to freeze it. We make it from a healthy donor, we freeze it, we store it, and then it can be administered to a patient quite rapidly. And in terms of the actual delivery of the therapy, it's quite straightforward as an IV infusion. So it's nothing complicated on that level. It's just there's a lot of complicated, or I'd say not complicated, but really brilliant science that goes in behind to produce the therapy.
And will the patients ever need like top up or maintenance treatments, or after the, let's say, 4 to 6 weeks and they've, eliminated all the cancer cells, is that it?
Yeah, again, this is a great question, and for those earlier therapies, it really can be one dose and that's it. And then the patients are cancer free. So what we're talking about with the iNKT cells, we still need to get that into clinic to see how patients perform. But we have the ability, if we needed to, that there's the opportunity to give multiple doses. So if the cancer was to return, you could consider doing that. But these are a lot of the questions that we answer as we progress through early stage clinical trials and beyond, and continue to, hopefully we can still continue to strengthen the product as well. And that's, again, more scientific experimentation.
And you were mentioning before that when it is really an aggressive cancer, it may be too late to do the treatment. So is there a particular stage of cancer that you know the therapy won't work for and they just shouldn't do it at all?
No, I don't think it's necessarily the stage. What we have is more specifically the cancer cell type. And so anecdotally, you know, I've spoken with an individual where they were told, you know, they're basically at that stage where the chemotherapy had failed and it was time, you know, to get their affairs in order. And fortunately, they were able to find their way onto a clinical trial for a cell therapy. And fortunately, this person's condition wasn't so aggressive that it was moving rapidly. But again, they took that therapy experimentally 4 to 5 years ago and they are still cancer free. So even when it's quite late stage, there's a possibility, but it's more about having the cancer type where those particular CARs will be able to find the cancer and kill it. And that's usually going to lead to better outcomes.
And when you guys have finished your clinical therapy, will the treatments be offered globally?
Yeah,
and
absolutely.
how do patients access it, I guess?
Yeah, excellent questions. And I think this is triggers a whole range of discussions. I think we need to do better to get people onto clinical trials and help them to understand what's available. But certainly for us, you look at the team that we've built. I think the last 4 out of the 5 recruits are all North American based. The technology is developed at Imperial College, so we do think very globally, and that's in the UK, I should say. We think globally. Of course, Australia is a wonderful place, I think, to test therapies and to do clinical trials, and we'd love to get the therapy to help Australian patients. That's really important. But in terms of where we'd be looking to target, in terms of territories to get the therapy out, it would be worldwide, ultimately.
And as in terms of revenue, would it work in terms of that you sell the technology to hospitals who offer it to patients?
No, it's oh, sorry, go on.
Sorry, yeah, no, you go on.
No, so it's essentially a patient cost and then reimbursement schemes that you would go through. But as I said, some of the therapies that are available today, they are in the order of $400,000 to $500,000 US per dose, which is, you know, when you equate that to saving somebody's life, it's probably not that much, but it's an enormous burden on individuals, healthcare systems that need, and insurers that need to reimburse and so forth. So I think the reality for us is that we just need to do better, so that we can reduce the cost of the therapy and get it out to more people. And that's where the off-the-shelf technology has a really big advantage, because you're not having to make the therapy for every single individual that's going to take it. You're able to make batches of the therapy and give that out when and as needed, which is, again, it's just going to reduce the logistical complexity of it.
And is there are there specific types of cancers that the therapy is more effective?
Yeah, excellent question. At this stage, it's blood cancers where we've seen the most effective display of the technology, and so they're things like lymphoma, leukemia, multiple myeloma, and they're where the drugs are already approved for. What we're always working on, though, is how to get these therapies and to develop them for use in solid tumours, because solid tumours do constitute a very large number of cancer cases. But to date, that's been a harder task, because the tumours actually surround themselves and create what's called a tumour microenvironment, where it's quite hostile. So things like CAR T cells and potentially CAR iNKT cells will have a harder time getting in. But I think that's where it really opens up the opportunity to work with not just one therapy alone, but combinations of therapies, so that we're tackling it on a multifaceted approach to hopefully yield better outcomes for patients. But certainly the success to date has been really predominantly in blood cancers.
And I'll take one more question. We don't want to take up too much of Michael's time. Do you think the market has hit a bottom, and how long do you think it will take to recover?
It's a great question. Look, and I'm not going to predict the future, so I can give some opinions, but certainly not financial advice. Look, these markets like this have typically lasted for about 12 to 18 months, and I think this downturn for the biotech sector has been happening since February last year. So we're well into it, but whether or not it's at the bottom, look, I think some signs look like things have stabilized a little bit, but I think with, not going off topic here, but worldwide there's still a number of headwinds, and inflation and still an issue, and obviously the war around the world. So there's a lot of factors that play into these things and how markets will respond. But certainly for us, it doesn't change the day to day. We need to get our skates on, hit our milestones, and get these therapies developed and as quickly as we can into patients so we can make a difference.
I guess it is on everyone's mind whether we have hit a bottom, but no one will ever know. But other than that, thank you so much, Michael. If you guys do have any questions, feel free to email me and I can pass them on to Michael. But it was a really great presentation. Thank you so much for your participation, and it was really insightful to learn more about Arovella. So thank you so much.
My pleasure. Thanks everybody for listening. Thank you for having me. Appreciate it.
Thank you.